Okay, I'm just looking here and they're making fun of Kennedy for claiming that he's going to be able to determine the cause of autism by September. And I say, why determine the cause? Why don't we just fix it by September? Can we do that? Maybe. Let's see what this guy has to say. I haven't watched this yet, but it's a short. It's only 2 minutes and 45 seconds. JFK claims he will determine the cause of autism by September. Well, let's see what he has to say. Here goes. So it's not fair for me to cut him off like that. I'm going to let him finish his statement. It's only a couple minutes. But Kennedy is saying we're going to put all these and we're going to do some testing. We're going to do some real research. And this guy said, oh, no, this is all bogus. So here we go again.
They're going to be about 1 in 31, 1 in 20, so they're going up again. From 1 in 10,000 when I was a kid. And we are going at your direction. We are going to know by September. We've launched a massive testing and research effort that's going to involve hundreds of scientists from around the world. By September, we will know what has caused the autism epidemic, and we'll be able to eliminate those exposures.
Hi, my name is Dr. Rubin, and what he said is bogus for many reasons. There's no way we're going to find out magically what are the underlying reasons why autism develops in the next 5 to 6 months. The scientific process doesn't necessarily have a hard timeline. It takes a long time to figure out issues. So first off, when we do scientific research, you have to get something called IRB approval when you are going to use human subjects, because you got to make sure that that research is ethically sound. And I've published research before and know that it takes months to carefully plan out the studies, write out your proposal, get funding, and get approval from your internal review boards of whatever institution that you are working with in order to actually move forward with that type of study. These types of studies that are going to be needed to look at a diagnosis like autism takes a lot of time to look at cohorts of people, to look at the potential exposures and what the eventual outcomes are years later. So it's going to take several years if you're going to do a high quality study to get meaningful results. And sometimes we don't necessarily get meaningful results, even if we are well intentioned and have well thought out studies. The causes of autism are most likely multifactorial, a combination of genetics and environmental factors. And also, we have had much more increased awareness about autism spectrum disorders, because back in the 1970s, 1980s, when my dad was a pediatrician, if you look at the pediatric textbooks that they would study from back then, autism was only a couple of paragraphs in those textbooks, and the diagnostic criteria were rather stringent. Compared to now, we have more awareness and the diagnostic criteria has broadened tremendously. My personal prediction is that if we're going to have any information by September, it is likely coming from studies that are highly flawed, low quality, and will not be reproducible by the scientific community. Because when we look at the underlying causes of diseases, we do need a scientific consensus where you can reproduce that data over and over again so that you can know what is that scientific truth.
Okay, that is the kind of response you get from academia. All I'm asking for, I'm not talking about an intrusive anything. I'm talking about bacterial swabs, just taking some poop. There's no ethical issue there. None, zero. And you see what's in there, because that is the output from your body. If you have the right bacteria in there, you have dissolved the things that come through your body. You have created the enzymes you need. If it's not coming out with the right stuff, if you got constipation, diarrhea, problems, gut distress, odors, all that stuff, it all points to a bad digestive process, which is 100% because you don't have the bacteria. That's my prediction. Now, maybe I'm wrong. But just because you've got a stethoscope hanging around his neck and a bow tie doesn't mean that he's right. And you say, oh, this is all bogus, let's just forget about the whole thing. No, we got to do the studies, my friend. Do bacteria in the body produce enzymes? Yes, in large amounts. It says bacteria in your body, especially in your gut, act like tiny factories, and many of them produce enzymes that are vital to your health. Let's clarify your questions, break it down. Do bacteria in the body produce enzymes? Yes, in large amounts. They create all the enzymes, basically. Gut bacteria produce enzymes that, let's see here, they break down proteins and fats, they digest, they build, they do everything in your body. They do every single thing. They do your immune responses. They're responsible for your, they live in these factories called the pancreas and the thymus and the liver, and they, inside of those factories, they do the chemistry that produce the enzyme, the chemicals that make your body run. Without enzymes, you would die almost instantly. All right, enzymes are the key. Are enzymes involved in nucleophilic substitution and invasion, which is how the living human body works? Every reaction basically is an invasion of another molecule, and it replaces something there. It's invasion and substitution. Now, I asked about enzymes. Are they involved? In biology, yes. In living systems, many nucleophilic substitution reactions are enzyme catalyzed. I say all of them are. Examples: DNA repair enzymes. They use nucleophilic substitution to replace damaged bases. They actually can repair DNA. Kinases like hexokinase. The catalase phosphate transfers via nucleophilic mechanisms. Phosphates are in your membranes. Serine protease use nucleophilic attack via a serine hydroxyl group to break peptide bonds. Everything is broken up and reassembled using enzymes. They work so fast that some of them do billions of years of chemistry in less than 1 second. That would happen, but it would take billions of years if you didn't have the enzyme. Billions of years. And in less than 1 second it happens if you have the enzyme. If you don't have the enzyme, it won't happen. Where did the enzyme come from? A bacteria. All right, we know bacteria are the factories that produce enzymes. We know enzymes do all the chemistry basically in your body that's fast enough to make your body work. So you kill your bacteria, what happens to your enzymes? You lose your enzymes. Well, what does that do? What happens if you have no enzymes in your body? You would die very quickly, within minutes to hours. It wouldn't take minutes. It would take probably 4 or 5 minutes. It's basically like you lost your breathing, because that pumps the blood through the body. Those enzymes are needed instantly throughout your body. Almost instantly, you can die from lack of enzymes. From everything: breathing, digestion, DNA, every single thing is affected when you lose your enzymes. So we know if you don't have your enzymes in your body, you're going to die within minutes to hours. Well, how could you lose your bacteria and then lose your enzymes? All right, like I say, I like fixing things, and I've been looking at this for a long time, and I believe what the fix is to replace the bacteria that's missing, so you get the enzymes, and then everything will clear up. Now, the kids can be repaired. This kid was 9.5 years old, nonverbal, never said a word in his life, and now he's virtually perfect, and he's more intelligent than most of his peers. All right, now, here's what I'm suggesting. We need a database of just taking fecal swabs. Very inexpensive, very easy to do, a small test group, and see the difference in the bacterial levels, what is missing or what is too much of. Then we can do a reasonable study of, can we replace these things and see improvement. Now, if all of that stuff works, we can use AI and spectral analysis to do this. Every single molecule has its own spectral signature. Virtually everything has a different spectral signature, which means it gives off a different a different amount of radiation in different frequencies, because the molecules bounce off radiation. So that's spectral analysis, very fully understood. Now, in addition to that, having that, to see the different colors and so forth of these different bacteria. You see the different colors? They know there's all these different colors. And so you could see it. You're going to be able to see it with a cell phone. Basically, you just take a picture of the poop. And if you see you got a bunch of the blue and you shouldn't have it, or you got a bunch of this looking color thing and you shouldn't have it, or not enough of that, whatever it is, send it into AI. Just go boom into AI. AI goes and looks at it, and then it also looks at this, and it says, "Well, you got all kind of problems with this or that. You got blah, 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 blah," and then it sends you back an analysis of your poop. Should be able to do it in a couple of minutes. Find out what's going on with the enzymes, because that's the bacteria. And AI should be able to do this, and you should be able to take it with a cell phone, send it off, and get the results literally just like you do now with chat. You go, and within a couple seconds, you got your results. I don't see this as a big do. I mean, I think it's simple. I really think it's simple. It's just been avoided and missed. And avoided, I think, because of the money. They're not going to be able to make a ton of money on this. This is natural products. Once you isolate the good bacteria, once you get the beneficial bacteria isolated, you just create them in vats. And people will be healthy, much more healthy than they are now, trust me. If you can adjust your bacteria levels in your gut, and you don't have distress, you don't have gas, you don't have bloating, you don't have diarrhea, you don't have constipation, you don't have acid reflux, you don't have nausea. Every one of those things is dysbiosis. That means your biotic in your gut is not working. It's called the brain gut something. It's the interface between your brain and your gut. Your brain has to have the chemistry or it can't work. Nothing works in your body. You get depressed. All kinds of things happen to you. Just from, and this is also hormonal activity. All of that stuff happens. And you need to see what's happening in your body because of enzymes. Hormones are nothing more than enzymes. And once you have those enzymes kicking around in your body, here's what happens. Those little ribosomes come out. They unsheath somewhere because they're needed. There's a trigger that triggers them. Bang. They go, bloop, and all of a sudden they go like this. Now, some of them come out as enzymes. Other bacteria make the same thing, work exactly the same way, but instead it makes a protein. Are they the same? Are enzymes proteins? Well, yes and no. They come from the same source of bacteria. They come out in the ribosome, yes. They unsheath the same way, yes. They do an opposite job. This breaks. An enzyme breaks things, destroys it. This builds it back up. That's a product. That's bone and muscle and tissue and eyeballs and everything else. And this takes the food you bring in and just smashes it to bits and says, here's all kinds of little bits and pieces. Do what you will with them. And the proteins, so if you can't break it, you can't build. You can't go and kill things that are trying to kill you. You become sick. It's a very simple process, and all we have to do is, like I say, get that bacterial database going. You know, it's very simple. It's a bacterial swab. It's very inexpensive, and you get a bacterial database of what's in these kids' digestive system. Without the enzymes, you're done. I'm done. All right, I love you all. We can do this. We can absolutely do this, and we don't have to wait till September either. All right. Primarily, we're talking about autism, but every single disease there is caused by some form of invasion because you can't stop it. How do you stop it? By these bacteria creating enzymes. Well, why would nuclear radiation sickness kill the bacteria versus you? And then you get stomach sick, you got throwing up, you get diarrhea and everything because your stomach and your whole guts are destroyed because you don't have any bacteria left really to create the enzymes. Without the enzymes, you're virtually dead within minutes. All right, you see what it says right here. Bacteria are usually more vulnerable. Why? Bacteria are single-celled. A single hit to their DNA kills them. They have no backup systems, no specialized repair tissue like humans do. We can fix ourselves. They can't, but you need enzymes to fix our cells. They're smaller, they're simpler, they're very small and very thin-skinned, and they are killed very easily. And without those, we die because we don't have the enzymes that they produce. All right, let's run through this. Very, very simple. This is what a bacteria looks like. Some of them have flagellas, some of them don't. Some of them have little fuzzy things, some of them have spikes, some have all kinds of things. But there is thousands and thousands and thousands and thousands of species in you. And every species has its own DNA. It's just, and then it has a little piece of that DNA says, I make a certain enzyme. Now, some of them don't make enzymes, bacteria. Some of them are cleaners. They go around and clean up all the goop in your body. They don't need to secrete enzymes. They do a different job. But the ones that do create enzymes, there's 75,000 enzymes or more in the human body. So there has to be that basically a number of bacterial species. Now, when you kill all those from nuclear radiation, done, and you are done. Almost instantly you start throwing up. Well, within an hour or two, you start getting sick, stomach sick, because this stuff is gone. It depends on how hard you got hit. If you got really whacked for a couple of minutes, within a couple of minutes you start getting really sick, because that's how fast these things do their chemistry. And you don't have it, you're done. You die within a couple of minutes without your enzymes. All right, so let's take this to its conclusion. What does the bacteria actually end up doing? It creates these ribosomes, these little tiny balls. And when I say tiny, these are tiny. You can fit 1 million of these little tiny balls in one single human cell. 1 million, even more. And then also you have all kinds of organelles in that cell, all kinds of things. And this is in there as well, 1 million of them. And they are ready to do their job instantly when called on. But you have to have the bacteria keep squirting them out continuously. And I know this because once your bacteria is dead, you get sick right away. So the ribosomes must be either getting killed at the same time, or they're, when I say killed, they're not alive. Ribosomes are not alive. They're just a chemistry set. Maybe it's getting broken up and destroyed. I don't know. But you don't have the enzymes. Almost instantaneously, you've lost your enzymes. Now, is it because the bacteria can't produce them? Or is it because the bacteria can't produce them and the ribosomes that should be floating through your body freely are also destroyed? This, I don't know. Nobody does. Nobody even knows about the bacteria and the enzymes and the correlation between that and the fluid-filled highway, which they claim was just discovered in 2018. That's this fluid-filled highway. Now, I discovered it myself, actually, and that's the truth. And ain't bragging if it's true. The paper's on academia.edu about fascia-facilitated fossilization. I found this layer in fascia in my fossils because it didn't squish. What happened was they never found this because by the time they did autopsies, it was flat. There was no more fluids in there. Well, my mud fossils, I could see the actual layer in there, and here's one right here. You see that layer? That's the fascia fluid highway. That's right in here. This was on an organ of some sort, I don't know what. But that's all the membrane on the top, that's the membrane on the bottom, and that's the fluid-filled highway that goes between them right here. Sorry, and I have the papers on the academia.edu. It's called fascia-facilitated fossilization. This is fascia. So at any rate, I've been studying this for many years, and I understand it quite well. And here's what happens. Those ribosomes squirt out. So here's the whole process all at one time, going up to fix DNA or change DNA, put in an immune signature up here. So what happens? Here's your bacteria. Yeah, it's doing whatever it does around the body, and it squirts out a ribosome. Okay? What does a ribosome consist of? It consists of a chain, a big long chain, or sometimes it's short. It depends on what it's doing. It can be extremely long. It's a polypeptide protein chains. Now these proteins are amino acids, and there's like 20 of them. And they have to hook in an exact perfect way, way long chains sometimes, and then they transcribe using transcription RNA, transferase it's called, which is an enzyme. It's an enzyme. This is an enzyme. Here's another enzyme up here, polymerase. So it shoots out all these balls, and they program an RNA messenger RNA strand, and that goes with uracil. See the U? The uracil snips into the DNA and inserts these CTAGs in your DNA, and it either fixes it or it makes you immune to something. It says recognize this. If you see this signature, kill it. And it sets up a factory to produce an enzyme to do that. Enzymes and viruses, bacteria and viruses, they change everything in your body. A virus isn't necessarily bad. A virus is nothing more than this. All right? It could be good, it could be bad. It depends on what it's going to make here or what it's going to break. That's all it is. A virus and bacteria's. Virus is the program. Think of it this way. Virus is a program. Once it gets into a DNA, it could into a bacteria, it could take over that bacteria and say, hey, I'm the new programmer here, everybody got out of the way, cancel that program, delete, here, put this program in. It starts squirting out ribosomes. Instead of making something good here, it starts making something bad, or it makes something better. Who knows? But the virus is the program. The bacteria is the worker. The bacteria does the job that the programmer tells it to do. It is literally that simple. And that, my friends, is the product of bacterial enzymes. They are so elegant. There is no way that you are going to get that chemistry unless you have that bacteria. It's just never going to happen. And it has to be identical to that every single time. Every one of these little balls has to be in the right order, at the right place, have its own magnetic domain. And then what it does is it goes around your body and it does what's called click chemistry. It's a magnetic executioner. Thousands and thousands and thousands of times, and it doesn't even get damaged. It continues to do what it does for I don't know how long, but for quite some time. And then you keep squirting them out. And these balls are in ribosomes. Before they turn into that, there's a ball so tiny, it can go through your blood-brain barrier and all that stuff. It's the smallest little particle you could ever imagine. You could put 1 million of them in one cell. There's no possibility that it couldn't transit the blood-brain barrier. There's no problem at all going through that. But once it opens up, no more going through the blood-brain barrier. So you have to put the ribosomes in there. That's the key. Right now they want to put drugs in there, and the drug is like this. It won't go through. You got a little thing like this. Well, this will go through that, and then it'll do this. But you can't do this and then go through the blood-brain barrier. That's the problem. So what is the way to approach this? We know there's beneficial bacteria, and we know there's harmful bacteria. And they live pretty much in your guts. And that transfers right into the lymph system, which is that fluid-filled highway they never even knew about until 2018. Most doctors still don't know much about it. And it is our immune system. So what happens is you got beneficial bacteria. We need to isolate that from stool samples. We need to find out what's good, what's bad. And the only way you can do that is to have a database. You take kids that are damaged and they have all kinds of whatever it is, because it's cancer, every single disease is invasion. It's invasion through a membrane, creating inflammation, creating cancer. Something else goes in and starts living on your vital tissue instead of you. But it's got to break through somewhere to get in. That's what cancer is. It's an invasion. So it's not only cancer that is affected by these lack of enzymes, it's autism, it's everything. And if you look up any single disease, I don't care what it is, and put it in associated to fecal matter transplant, they will say, ooh, it looks good, but we need more work. Ooh, it cured this and it cured that, but we need to do more research. Ooh, this and ooh, that. And if you look it up and say, what did the government do? They shut the whole thing down, basically. The company was going to do it was Open Biome, and they wanted permission to start doing this stuff, and the government said no. And they finally gave up to trying to get this certified. And it really is wrong to not allow this, because once they can isolate the bacteria, they could just create them in vats. Problem is, you can't create money from this. You can't create gigantic amounts of money because you can't patent bacteria. You can't. It's a natural thing. You can't patent something, a living product. It's just living. You can't take it and say, that's mine, I own that. No. So what they want to do is do some little alteration to it, which makes it not really the right bacteria, but as you can patent it because you said, oh, I altered that, I own it. That's what the whole deal is, and that's why we're never getting anywhere with getting diseases repaired, because there's not enough money in it.